Yes, I welcome you all on Dr. Reena's Physiology, your faculty, your mentor. And as you can see, the topic today that we will be discussing will be nothing but a beautiful synapse, right? Now, what is synapse and why we need to study this? See, I am communicating with you through this OSB and through the studio and getting you a lecture. Similarly, we need to speak to each other to maintain coordination. That is the main function of brain, that is coordination. So nervous system का सबसे important function होता है coordination. इस coordination के लिए what is very important is communication. You know, communication skill तो even we have to learn as being one of the best Indian medical graduates, or the IMG which we call. So the brain is communicating with each other, with the different neurons, to bring about coordination. One of the communication we've already seen, that how the neuron communicates with an effector organ, that is the skeletal muscle, through the NMJ. And therefore the first lecture which I took was NMJ, neuromuscular junction. Now today we will learn how do the neurons speak to each other, right? How do the neurons speak to each other through the way of this physiological communication, which we called as synapse, right? So to bring about effective coordination, there is need of communication, and that communication is brought about by synapse. You can log on to this Telegram group, because this entire lecture will be converted into PDF and you can get the notes of it. The must known competencies, okay, the must known competencies of this topic are: you should be able to classify synapse, draw a well labeled diagram, understand the synaptic potential, what will be the difference between EPSP, IPSP, what will be the difference between the electrical synapse and the chemical synapse. Okay, so what is actually a synapse? Remember, it is a way of communication. It is one way flow of information. It is a one way flow of information between two neurons, right? Or it is there is no anatomical contiguity between the neurons, right? There is no anatomical contiguity between the neurons, but they are connected to each other by this functional way, which is synapse. So you can define it that it is a site where action potential travels across a gap. Okay, action potential travels across a gap that is 20 to 40 nanometers wide, which we called as the synaptic cleft, and this is going to separate the axon terminal, this axon terminal, which will be called as the presynaptic terminal, from the dendrite, or from the soma, or from the axon terminal of another synapse or another neuron, which we will call as postsynaptic, right? That means the axon terminal can form a synapse with the dendrite or with the soma or with the axon, right? Now very interestingly, you must remember that there are about 10 to the power 12 neurons in the brain. Okay, there are so much neurons in the brain, and about 10 to the power 15 synapses. That means you can imagine the complexity of the neural circuitry with so many synapses. And कैसे होता है? One of the neuron, it may receive synapses from so other neurons because of the process of summation, which we will see later on. So let's look into one MCQ before I take you forward towards this topic. A 48-year-old woman who is multiple sclerosis, having increased spasticity, is treated with an intrathecal infusion of baclofen. What is baclofen? It is an analgesic, which is actually a GABA agonist that mediates presynaptic inhibition. So we are going to study this topic later on. What is presynaptic inhibition, postsynaptic inhibition, and so on. This presynaptic inhibition is brought about in the CNS, which is going to affect the firing of the alpha motor neurons. You have to tell me by which mechanism. Options: in decreasing the frequency of action potential of the presynaptic nerve ending, decreasing the potassium permeability of the alpha motor neuron, hyperpolarizing the membrane potential, increasing the amount of neurotransmitter released, or increase the chloride permeability at the presynaptic nerve ending. So what is the mechanism of action of this GABA? Is E, that is, it will bring about increase in the chloride permeability. So by increasing the chloride permeability, right, this increase in the chloride conductance is going to decrease the magnitude of action potential. That means the action potential which is going to be conducted from the presynaptic to the postsynaptic neuron is going to be affected by an inhibitory neurotransmitter, that is GABA. और कैसे? By increasing the chloride conductance. So let's get on, boys and girls, to this very important topic. So as I told you, synapse, it is a physiological union without any anatomical union, and there are about 10 trillion synapses in the brain. How can you classify them? This comes as a 2 marks BAQ. So you can classify them in two forms. One, which is called as the anatomical classification. Second, which is physiological classification. So physiological classification can again be based on the type of transmission or based on the type of response. So anatomical classification, it can be exodendritic, that means axon forming a connection with the dendrite. It can be exosomatic, that means axon forming a connection with the soma, that is the body, or exoaxonal, which is the rarest form, okay, which is the rarest form of a synapse, right? So first important anatomical classification is exodendritic, exosomatic and exoaxonal. Abhi based upon transmission and based upon the response. So based upon the transmission, मतलब transmission कैसे हो रहा है? It can be a chemical synapse or it can be electrical synapse. So physiologically you can classify them as two types, electrical, chemical, ठीक है, and then based upon the response which we will see. सेकेंड होता है बेस्ड अपॉन रिस्पॉन्स, बेस्ड अपॉन ट्रांसमिशन देखा हमने कि इलेक्ट्रिकल या केमिकल, और रिस्पॉन्स के हिसाब से यू कैन फर्दर क्लासीफाई देम, आइदर इट कैन बी एक्साइटेटरी, ठीक है, इट कैन बी एक्साइटेटरी विच विल बी EPSP विच वी विल बी स्टडिंग, और इट कैन बी इनहिबिटरी विच इज कॉल्ड एज IPSP, तो ये भी हम पढ़ेंगे. सो हमारा 2 मार्क्स का PAQ कवर हो गया. टाइप ऑफ सिनैप्स, एनाटॉमिकल एंड फिजियोलॉजिकल, अगेन 2 टाइप्स, ट्रांसमिशन के टाइप से एंड रिस्पॉन्स के टाइप से, ठीक है. नाउ दिस इज विच टाइप ऑफ सिनैप्स? दिस इज अ केमिकल सिनैप्स. ये केमिकल सिनैप्स क्यों है? व्हाई एम आई कॉलिंग इट एज अ केमिकल सिनैप्स? You can see that the chemical synapse is just analogous to that of your NMJ, because यहाँ पर there is going to be release of a neurotransmitter. This neurotransmitter after being released will bind to its site on the postsynaptic membrane, which could either open the channels, which could be either inotropic. So inotropics जो होते हैं ना, they open the channels, या it can be मेटाबोट्रोपिक. मेटाबोट्रोपिक वो होते हैं जो कोई सेकेंड मैसेंजर यूज करते हैं. सेकेंड मैसेंजर इज साइक्लिक AMP, साइक्लिक GMP, कैल्शियम, ठीक है. सो न्यूरोट्रांसमीटर का एक बहुत इंपॉर्टेंट रोल है कि बिकॉज इट इज और केमिकल सिनैप्स का एक बहुत इंपॉर्टेंट ट्रांसमिशन है कि इट इज ऑलवेज वन वे कंडक्शन. दैट मीन्स ऑलवेज फ्रॉम द प्री सिनैप्टिक टू द पोस्ट सिनैप्टिक. क्यों ऐसे होगा? बिकॉज जो केमिकल है वो कहाँ से रिलीज होगा? प्री सिनैप्टिक से, और बाइंड कहाँ पे होगा? पोस्ट सिनैप्टिक में. सो इट कैन नॉट बी उल्टा, पोस्ट से प्री कभी नहीं जाएगा. इट विल ऑलवेज बी फ्रॉम द प्री टू द पोस्ट. So this is the chemical which is brought about by neurotransmitter. And now you look upon to the electrical synapse. अब आप देखोगे electrical synapses में gap junctions हैं. So these are, when we learn gap junctions, we call them as cytoplasmic channels. तो जो gap junctions होते हैं, they are basically connecting the cytoplasm or the exoplasm of the two neurons and this is connecting the two neurons with the help of this gap junctions. However, it can also be bidirectional, इधर से उधर या उधर से इधर, it can flow. So there is no chemical neurotransmitter which is involved, right? Now, after being understanding nicely the types of synapses, next very important thing is to know the structure of the synapse. Understanding the structure of the synapse is not very difficult now because you've already studied NMJ, that is the neuromuscular junction. So आप याद रखो that this was your neuron or cell body. This was the cell body which we called as soma, right? The dendrites were there. These were the dendrites. These dendrites were nothing but the receptive region, जहाँ पे you can see the local potentials will be generated. This portion is the initial segment, and initial segment के बाद में there is going to be the axon hillock and initial segment. This conical portion is the axon hillock and then the initial segment, right? This is going to be the axon, which is now going to end in the form of the nerve terminals, which will have this synaptic knob, and this synaptic knob will have the neurotransmitter substance. So the local potentials, they are received by these dendrites. They are summated in the soma, and the initial segment and the axon hillock, they are supposed to be the site of generation of, they are the site of generation of the propagated action potential. इसका मतलब summation के बाद में जो हमने action potential create किया है, that will be propagated from that site. क्यों? Because this initial segment and axon hillock, they have got least threshold for excitation. They have got least threshold for excitation because there is a very dense amount of sodium channels which are present there. So physiologically, the initial segment and the axon hillock are very important because they are the site of generation of the propagated action potential. And if the nerve is myelinated, then there will be nodes of Renvir and you will see the saltatory conduction. Now we are looking on over here, that is this terminal portion or the synaptic knob, which is rich in mitochondria, which tells you that there is a very high metabolic activity. Synaptic vesicles, which will be loaded with the neurotransmitter, and this is the vesicle docking site. We've already seen this in the NMJ, and they're going to be calcium channels over here so that the calcium can come inside and calcium can bring about the release of the neurotransmitter. On the postsynaptic cell, a very important thickening of the membrane is called as the postsynaptic density, because this postsynaptic density will have the receptors where these released neurotransmitter are going to bind, right? So you're going to have the presynaptic terminal, you're going to have the postsynaptic terminal, you're going to have this space, and we know that this space is nothing but what we called as the synaptic cleft, which will have ECF, it will have the enzyme that may result in the destruction of the neurotransmitter, it will have calcium. Right? So presynaptic ending, synaptic cleft, and the postsynaptic ending, very important, the postsynaptic density. Now one thing which you can remember, so we've considered that there is a presynaptic terminal, postsynaptic terminal, synaptic cleft. A cheese at the extra edge you can remember is something which is called as the neurexins. These neurexins, they actually connect the presynaptic terminal to the postsynaptic terminal, right? They connect the presynaptic terminal to the postsynaptic terminal. They are not only important for holding the synapses, okay? They are not only important for holding the synapses or providing strength, but very importantly, they have synaptic specificity. That means a particular neurexin can bind to a particular receptor on the postsynaptic side. So neurexin is an extra edge which you can remember for yourself. Now next is This is again a very important 2 marks BAQ, that is what is the difference between chemical and electrical synapse. So we've seen that the chemical synapse do not have any anatomical contiguity. The release of the neurotransmitter is going to be there from the presynaptic terminal, which will end on the postsynaptic, and then you will be getting the potentials. And because the release of the neurotransmitter is from the presynaptic terminal and the binding is on the postsynaptic terminal, ये वाला जो synapse है ये हमेशा unidirectional रहेगा, मतलब हमेशा from pre to post. Electrical synapses are because of the gap junctions, and the impulse transfer can be bidirectional. And through this gap junctions आप क्या करोगे? What are you going to do? Direct impulse transfer from the presynaptic to the postsynaptic with the help of ions, right? No neurotransmitter. Now this chemical synapse, इसका जो synaptic cleft होता है थोड़ा बड़ा होता है, 20 to 40 nanometers. Electrical synapse, इनमें synaptic cleft some of them may not have even, और जिनमें होता है it is very small, around 2 nanometers. Now जब आप repeatedly chemical synapse को stimulate करेंगे तो क्या हो जाएगा? There will be exhaustion of the neurotransmitter substance, and because there is exhaustion, that means they are vulnerable to fatigue. Okay, they are vulnerable to fatigue. In comparison, electrical synapse is less susceptible to fatigue. So very very important difference between the chemical and the electrical synapse, which is my 2 marks BAQ. So if I am an examiner, I can really put this because you can write it very well. Now come to how is the synaptic transmission taking place. इसको समझने में अभी कोई भी प्रॉब्लम नहीं आएगी बिकॉज हमने ऑलरेडी NMJ कवर किया हुआ है फर्स्ट लेक्चर में. नाउ सी the action potential travels along the axon and reaches the terminal, मतलब लास्ट सिनेप्टिक नॉब जहां पर है वहां पर वो रीच होगा. ठीक है, जैसे ही वो रीच हुआ इट इज गोइंग टू ओपन द कैल्शियम चैनल्स. सो एक्शन terminal depolarization. This depolarization is then going to result in the opening of the calcium channels and calcium अंदर चला गया. This calcium is then going to bring about the release of the vesicles by exocytosis. This acetylcholine which is released will bind to its ligand on the postsynaptic membrane. So at the level of NMJ it is हमेशा acetylcholine, लेकिन acetylcholine अलग जगह पर न्यूरॉन्स पर भी हो सकता है. एंड इफ यू रिमेंबर, जो इसका रिसेप्टर था हमने पढ़ा था, इसका रिसेप्टर होता है एक पेंटामर दैट इज हैविंग 5 सब यूनिट्स, 2 अल्फा, 1 बीटा, 1 गामा एंड 1 डेल्टा. जैसे ही दिस एसिटिलकोलिन विल बाइंड टू इट्स रिसेप्टर, sodium influx will take place and you will develop a local potential. Synaptic potentials are local potentials, right? And then they will be converted into an action potential. And we know that once the acetylcholine has done its work, it will be then destroyed or degraded by the enzyme acetylcholine esterase. So this is the synaptic transmission event. So I have made this for you in the form of a flow chart. मैं हमेशा बोलती हूँ सब बच्चों को कि जब भी आपको एग्जाम में आंसर आता है फिजियोलॉजी में, 2 तरीके से यू कैन मेक योर लाइफ इजी, either by diagrams or by flow charts. If you put this answer in the form of a flow chart, you bang on, I'm going to give you full marks, right? So the impulse arriving, depolarizing the presynaptic membrane. Depolarizing matlab making it less negative, like between -40 to -20. This depolarization is responsible to open the calcium channels in the presynaptic membrane. ये बहुत important है कि हमने calcium channels को कहाँ पर खोला है? Presynaptic membrane में, resulting in the influx of calcium. calcium stimulating the exocytosis of the vesicles. The vesicles are then going to release the neurotransmitter, that is acetylcholine, and if the current is depolarizing, it will be an excitatory potential. Something very, very interesting. The release of this calcium is actually a graded response, right? It depends upon the frequency of action potential. If you increase the frequency of action potential, what will happen? Suppose you increase the frequency of action potential. So if I increase the frequency of action potential, the amount of calcium released will be increased. If the amount of calcium is increased, the amount of neurotransmitter is increased, and more response at the level of postsynaptic membrane. So this feature, boys and girls, this feature is very, very important, that increase in the calcium release, which will cause increase in the release of neurotransmitter from the postsynaptic membrane, and more of the response. This is what is called also as post-tetanic potentiation, also called as long-term potentiation, and these are very, very important processes which take place in your memory and learning at the level of hippocampus. So ये जो graded response है, if you keep on increasing the release of calcium by increasing the frequency of action potential, the response at the postsynaptic membrane will be more, and this is how you convert the short term memory into a long term memory. तो इसको अभी यहाँ पर ही आप समझ लो, so it becomes very easy. Now अभी देखो हमने क्या किया, we brought about the release of an excitatory neurotransmitter. मतलब जो neurotransmitter release हुआ, forget acetylcholine, that is at the level of NMG. Suppose हमने एक excitatory neurotransmitter release किया, जैसे glutamate. So this glutamate is an excitatory neurotransmitter. What it will do? It will increase the sodium influx, है ना? तो जब भी आपको एक्साइटेशन लेकर आना है या डीपोलराइजेशन करना है, तो आपको क्या करना पड़ेगा? आपको इनफ्लक्स करना पड़ेगा पॉजिटिव आयंस का. यू विल हैव टू इंक्रीज द इनफ्लक्स ऑफ पॉजिटिव आयंस, जैसे ज़्यादा सोडियम या फिर कैल्शियम. एट द सेम टाइम क्लोज द पोटेशियम चैनल्स. So the second way of depolarizing is close the potassium channels. If you close the potassium channels, what will happen? There will be no efflux of potassium, and if the potassium remains inside, again there will be depolarization. That means the RMP which was 70, you took it up, made it more and more positive, or you made it less negative. आपने क्या किया? उसको less negative किया. So when you are decreasing the negativity, it is nothing but EPSP, that is excitatory postsynaptic potential. Now if you release an inhibitory neurotransmitter, for example glycine, for example GABA, these inhibitory neurotransmitters, they don't cause depolarization, but they cause hyperpolarization. They increase the negativity. So increase in the negativity can be brought about either by bringing inside negative ions like chloride, or throwing out positive ions like potassium. So potassium is already more inside, उसको आप बाहर निकाल दो, नेगेटिव हो गई, या आप अंदर नेगेटिव आयंस ले जाओ. सो आपका जो RMP -70 था, उसको आपने और नेगेटिव बना दिया. सो व्हेन यू आर मेकिंग योर RMP मोर नेगेटिव, दिस इज व्हाट यू आर डूइंग, दिस इज व्हाट यू आर डूइंग टू ब्रिंग अबाउट हाइपर पोलराइजेशन और इनहिबिशन ऑफ रिस्पॉन्स. सो रिस्पॉन्स पोस्ट सिनेप्टिक में पॉजिटिव होगा या नेगेटिव होगा, दैट इज गोइंग टू बी डिपेंडेंट टोटली अपॉन द आयंस विच आर गेटिंग influx or efflux, right? So देखो अब आप just understand through this figure to become more easy to you. This is nothing but this is an inhibition of impulse. So you brought about release of an inhibitory neurotransmitter from these vesicles. Again, the release of the vesicles is always by calcium from the presynaptic membrane. This inhibitory neurotransmitter, it brought about binding. Once this inhibitory neurotransmitter binded to its receptor on the postsynaptic membrane. So it is ligand gated channel because a ligand, that is the inhibitory neurotransmitter, is binding. इसने क्या किया? It resulted in opening of the calcium and increase in the calcium influx, and the impulse is not conducted because it was inhibitory postsynaptic potential, which made it more negative. That means you brought about nothing but hyperpolarization of the postsynaptic membrane. You did not allow the impulse to be conducted. ठीक है, अगे ये आप लोगे excitatory. Again, nerve impulse traveling to the presynaptic terminal, opening of the voltage gated calcium channels, this calcium helping in the exocytosis. You are bringing about this binding of the excitatory neurotransmitter on this postsynaptic membrane. This time this is causing sodium influx, that means positivity inside, leading to an excitatory postsynaptic potential. Now the impulse will be conducted in the form of action potential. So this becomes my excitatory postsynaptic potential. So the potential that develops at the synapse is called as a synaptic potential if it is excitatory or if it is inhibitory. Next important 2 marks BAQ. What is the fate of the neurotransmitter that is released? आपको पता होना चाहिए. This can be enzymatically degraded. For example, acetylcholine was destroyed by acetylcholine esterase, right? So you know that acetylcholine can be degraded by the enzyme acetylcholine esterase. Similarly, catecholamines, epinephrine, norepinephrine, they are degraded by COMT, that is catechol O methyl transferase. Catechol O methyl transferase for your catecholamines. Some of the times there is reuptake of the neurotransmitter by the same nerve ending that have released it. Some of the time it is just removed by diffusion, or in the case of central nervous system, the neurotransmitters, they are removed by astrocytes. So this can also come as a 2 marks BAQ. What is the fate of the released neurotransmitter? This is one of the very favorite BAQ. What is the difference between excitatory and inhibitory SPs? So we've seen it is brought about by a excitatory neurotransmitter like glutamate with either sodium influx or calcium influx, or you can bring about the closure of potassium channels. The positivity which it creates is about +8. That +8 matlab more towards positive side comparing to the RMP, with a latency of 0.5 milliseconds, rising to a peak in 2 milliseconds and declines with a half-life of 4 milliseconds. IPSP is brought about by inhibitory neurotransmitter, that is glycine or GABA. It either brings about potassium efflux or brings about chloride influx. So potassium efflux matlab positive ions will go out. The potential is around -2 millivolts, latency 2 millisecond, and returning back towards RMP with a half-life of 3 milliseconds. So in one word you can remember that this is going to be depolarizing and this one is going to be hyperpolarizing. Itna dhyan rakho. Now, where do you bring about this action potential? The first thing which you do is the synaptic integration. The dendrites is going to receive all the synapses. Some of the synapses can be positive, inhibitory, some of them can be excitatory. You will have to bring about an algebraic summation. जैसे हम algebra में करते थे plus minus, तो इनका summation होगा. So this is the phenomenon of summation at the postsynaptic membrane, which is called as the synaptic integration. The synaptic integration, that means it is summation, and we will see what is summation. It is temporal summation, it is spatial summation. We will study this when we study the properties of synapse. Okay, so it is the net algebraic summation of the different potentials which we are receiving at the postsynaptic membrane. Then you will develop an initial spike. So if the summated postsynaptic potential is excitatory, it will depolarize the initial segment by 30 millivolts. This initial spike will trigger the action potential, which will travel in the antegrade direction. That means it will always be from the presynaptic to the postsynaptic. It will travel from the axon towards the terminal. So in the antegrade direction. But a very small amount, okay, a very small amount of a retrograde spike is seen. Retrograde spike, that is a somatodendritic spike. That means it will go back towards the soma and the dendrite of the presynaptic neuron. So this was the presynaptic neuron, this is the postsynaptic neuron. This is antegrade from the presynaptic to the postsynaptic, but thoda sa jo direction hai, right, thoda sa direction, it is like this, ulta. That means, okay, again see, what did you do is you generated impulse, okay, and this is you conducted. This is how you conducted the impulse. This is antegrade, but a small amount will be like this. That is somadendritic. This somadendritic is very, very important, because what it will do, it will restore the soma and the dendrites back to resting potential. So you are taking antegrade, conducting it forward, but small amount of retrograde, that is towards the soma and the dendrite. This is called as retrograde somadendritic spike, which will restore the potentials which were accumulated over here. For example, I am cleaning this space now. Why am I cleaning this space? Because I want to write more, or I want to draw another one. So ये next draw करने के लिए जो पुराना था हमने clear कर दिया. The same thing is what is the somadendritic retrograde spike. You want to clear the potentials, local potentials, and now you want to get ready for the next one. Now, another very important question, 2 marks BAQ, properties of the synaptic potential. Synaptic potential is not a true action potential. These are local or the graded potentials which show summation. Again, we will learn that summation is of 2 types. It is either a temporal summation or it is either a spatial summation, and they do not obey all or none law. They can also be decremental. Decremental, that means they can even decline. So they will have to cross the threshold of excitation to get converted into an action potential and propagate. Till the action potential is not generated, they can be IPSP or EPSP, and these are graded potentials. They are not true action potentials, right? Finally, what are the factors affecting synaptic transmission? Again, important BAQ. First one, botulinum toxin. Remember, botulinum toxin I have also covered in NMJ, which prevents the release of acetylcholine and causes flaccid paralysis. It causes flaccid paralysis. This was the action of the botulinum toxin because it interferes with the function of the docking proteins. This I have already covered in the lecture of NMJ. The second important example is the tetanus toxin. अगर आपको याद होगा हमारे टेलीग्राम ग्रुप पर मैंने इसका MCQ दिया था, that tetanus toxin, it causes presynaptic inhibition and it prevents the release of inhibitory neurotransmitters like glycine and causes lockjaw like spastic features. Next important is your acidosis, which is known to suppress the synaptic transmission by decreasing the release of neurotransmitter. Alkalosis, which facilitates the synaptic transmission because it increases the release of the neurotransmitter. On the other hand, hypnotics, anesthetic agents, they all are known to depress the nerve conduction and therefore they decrease the synaptic transmission. On the other hand, caffeine, nicotine, even tea, what do? They decrease the threshold for excitation, facilitate transmission. When we are studying, we take a cup of tea. That tea is not required for the body, it requires for the mind because it is going to cause stimulation of the synapse. And we also teach you, boys and girls, if you remember, that the seat of fatigue in human beings is synapse. जब हम NMJ का practical पढ़ते हैं fatigue में, तब आपको बताते हैं कि that the seat of fatigue in human beings is synapse. So a small motivation, right, a positive stimulant like a cup of coffee, they are all increasing our synaptic facilitation or transmission. Opioids, they also inhibit the synaptic transmission. right? Now this is an extra edge if any of my learners are very much, you know, keen to learn and keen to learn the extra knowledge. This is for you guys, that how does calcium releases neurotransmitter. This is an extra edge for you. When the calcium is coming into the presynaptic membrane by the opening of the voltage gated channels, the calcium activates the calcium calmodulin dependent protein kinases. Once there is activation of the protein kinases, they are going to phosphorylate a protein which is called as synapsin. This protein synapsin is actually associated with the synaptic vesicles, and once it gets phosphorylated, it will disassociate from the vesicles. Once the synapsin disassociates from the vesicles can move towards the membrane and they will fuse with the membrane, that is the presynaptic membrane, to be released by exocytosis. Very interestingly, this process requires cytoskeletal proteins like actin. That means जितना काम हमें contractile proteins का होता है in muscle contraction, उतना ही काम हमको होता है vesicles को उनकी जगह से लेकर जाओ towards the presynaptic membrane and release them. So microtubules, cytoskeletal elements like actin are very important contractile proteins even for exocytosis. So calcium calmodulin kinases, phosphorylation of synapsin and release by exocytosis. So boys and girls, this is all for this lecture. I just tell you all that always make your learning a very happy learning. Learn physiology in the way you are learning or reading a story. Don't mug up, because this is very important core subject for your medicine, pharmacology, your pathology, or whatever you learn in your near future. Please get connected to the channel, subscribe the channel, let more and more people learn physiology, because I know that I put my best efforts. These notes are the notes which will give you a grade of more than 75%. I assure you, being an examiner, that my notes are going to make you score more than 80%, right? So these will be available on the Telegram group. Share the channel and please put a comment so that I come to know what is the way of teaching. Thank you all. Have a blessed day.